Bismillahirrahmanirrahim. I am Niamatullah, student of BS7, Department of Microbiology, Cohort, University of Science and Technology, Cohort. Today my presentation topic is determination of resistance lactobacillus to low pH and bile salt. So now we will discuss about how lactobacillus respond to low pH and bile salt. During literature review about my presentation topic, I found this research paper which is about evolution of resistance to low pH and bile salt of human lactobacillus species isolate, which is published in a very reputed journal named International Journal of Immunopathology and Pharmacology. Now I'm going toward content of my presentation topic. Following are the main content of my presentation topic, that is introduction, objective, material, methodology, research, and at last reference. Now introduction. Probiotic are mostly normal flora. As we know that lactobacillus are probiotic. Probiotic mean that probiotics are basically microorganisms introduced into the body of human for its beneficial quality. Probiotics are live bacteria and yeast. Probiotics are often called good or helpful bacteria because they help keep your gut healthy, especially digestive system. Probiotics also have pathogenic nature and cause disease when they change their site. Most common two groups of probiotics are Lactobacillus and Bifidobacterium. But, but in this lecture, we will discuss Lactobacillus. Now I am going to discuss Lactobacillus. What is Lactobacillus? Most common probiotic. Lactobacillus is a genus of gram-positive. Lactobacillus is aerotolerant, anaerobic, or microaerophilic. Aerotolerant means that they can survive without using oxygen because they obtain the required amount of energy through fermentation. Aerophilic means that which require oxygen for producing energy in the form of ATP. Lactobacillus is rod-shaped non-spore-forming bacteria, habitat in yogurt and other fermented foods. It means that Lactobacillus is present in yogurt and other fermented food. Different strain of lactobacillus can help with diarrhea and may help people who can't digest lactose, the sugar and milk. Lactose is sugar and milk. Major part of lactic acid bacteria group. Lactobacillus is the major part of lactic acid bacteria group. It converts sugar to lactic acid. It means that they basically convert lactose into lactic acid by using oxidized NADH, which produced or generated during glycolysis. Lactobacillus resist to low pH and bile salt. Now objective. The main objective of this topic is, is given below. Probiotic prevent the development of wide range of human and animal pathogens such as Staphylococcus aureus, Bacillus subtilis, and Pseudomonas aeruginosa. As we know that Staphylococcus aureus cause skin infection. Bacillus subtilis cause infection of ear, eye, and respiratory tract. Pseudomonas aeruginosa cause infection in the blood, lung, or other part of the body after surgery. The main aim of this study to investigate the lactobacillus, mainly lactobacillus acidophilus resistance or tolerance to low pH and bile salt. The main aim of this study how lactobacillus resist or tolerance to low pH and bile salt. Optimum condition for growth. How lactobacillus grow, which temperature required for lactobacillus, uh, optimum temperature for lactobacillus is 37 degrees centigrade as our body, human body normal temperature, which is 99 degree Fahrenheit. Optimum pH is 5 to 6 degrees centigrade, which is low pH. Material. For the determination of resistance lactobacillus to low pH and bile salt, we will use the following material, that is commercial yogurt, ethanol, microscope, 10 ml glass beaker, pH 100 ml glass beaker, pH indicator paper, glass spatula, 1 mole HCl or 1 mole NaOH, vial salt solution, test tube, burner, microscope slide, MRS agar plates, pH meter, inoculation loop. Now, how we can identify Lactobacillus acidophilus? For the indication of Lactobacillus acidophilus, we will use the following test, that is gram staining, catalase test, carbohydrate fermentation test, methyl red test, VP test. After doing the above test, following results come which are shown in this graphs. We will done the above test for the following three species of Lactobacillus acidophilus, that is lactobacillus only, Lactobacillus acidophilus, Lactobacillus casei, Lactobacillus lactus. When we do these three tests, when the shape of these three species are bacilli, it means that the shape of these three species are rod. Catalase tests for these three species are negative. Motility tests for these three species, that is Lactobacillus acidophilus, Lactobacillus casei, Lactobacillus lactus, are also negative for these three species. Growth at NSL are positive for these three species. Methyl red tests are positive for these three species. Rheas tests are also positive for these three species. Lactose tests are positive for these three species. Mannose tests are positive for Lactobacillus acidophilus, negative for Lactobacillus casei, or positive are also positive for Lactobacillus lactus. Repanose tests are also positive for these three species, and etc., which are shown in these graphs. Now, how can we determine the resistance of Lactobacillus acidophilus to low pH? Demand, rugosa, and sharp MRS broth was prepared with different pH. First of all, what is MRS broth? MRS broth originally developed in 1960 by the man Rugosa and Sharp. MRS broth is a medium for the cultivation of Lactobacillus species. The man Rugosa Sharp MRS broth was prepared with different pH. that is 1, 2, 3 by adding 1 mole HCl and 10 ml 3 different test tube. 1 mRS broth test tube with 5 pH served as a control sample. We take 1 mRS broth test tube with 5 pH as a control sample, which act as a control sample, and then take, take a colony of Lactobacillus acidophilus with inoculating loop and then emerge and these test tube separately. And then incubate at 37 degrees centigrade for 24 hours. We incubate it for 37 degrees centigrade at 37 degrees centigrade for 24 hours. Bacterial growth is monitored by measuring absorbance with a spectrophotometer at 600 nanometer after 4 hour 4 or 424-hour pH, also measured with pH meter or pH indicator. It means that we will keep observing the growth of bacteria and will measure it through spectrophotometer at 600 nanometer after specific interval of time according to the mentioned time. MRS broth is prepared with different concentration of bile salt at 0.1, 0.3, 0.5 and 1.0% as dispensed and 10 ml volume test tube and sterilized at 121 degrees centigrade for 15 minutes. After that, 1 mRS breast test tube with 0% bile salt served as a control sample. Here, 0.0% bile salt used as a control group. A colony then again take a colony of Lactobacillus acidophilus with inoculating loops and then immerse and these test tubes separately. Again incubate at 37 degrees centigrade for 24 hours. Bacterial growth is monitored by measuring absorbance with a spectrophotometer at 600 nanometer after 4 hours or 24 hours and pH also measured with pH meter or pH indicator. After the completion of the experiment, we will observe the following results which are shown in the figure, that is 1.0, 2.0 pH, 3.0 pH, and 5.0 pH. From this graph, we conclude that as the pH of the body decreases, Lactobacillus acidophilus show resistance to that. Discussion. The viability of Lactobacillus acidophilus decreases significantly. After incubation at pH 3.0 below pH 3, the number of bacteria in the medium decreases because the loss of viability. At pH less than or equal to non-viable bacterial cell were detected of the first hour, suggesting most isolate were killed by severe pH. Severe pH means high pH. Lactobacillus acidophilus show reasonable growth at all bile salt concentration study. The bile tolerance results were 87.41%, 75.49%, 69.53%, and 54.96% for the 0.1%, 0.3%, 0.5%, and 1.0% of the bile salt concentration. The ability of the Lactobacillus acidophilus to resist bile salt. This shows that Lactobacillus acidophilus resistance decreases by increased bile salt concentration. But some strain of Lactobacillus which are written here are resistant for 3.0% bile concentration. This is the reference of my topic. Any question? Thank you.